Hi, everyone. Welcome to this new episode of our Student Spotlight series, where we're able to showcase some of the work our past students have done. I'm Charlotte. I'm one of our outreach coordinators here at CCIR, and it's our pleasure to have with us today, Aadi. So can you just introduce yourself for us? So tell us where you're from, what year you are in school, and then a little bit about the course that you took.
Yeah. Well, first of all, it's an honor to be here. I'm really happy to be a part of the CCIR Student Spotlight. My name is Aadi Desai, and I was a student at Navrachana Higher Secondary School, and I recently passed my 10th grade on my sophomore year. And my course was Cancer Bioinformatics, Mining the Big Data, which was taught by Professor Dr. Begum Akman from the University of Cambridge.
Great. And then just to confirm, that was one of our Future Scholars Program courses. Is that right? Great. So for people who aren't familiar with bioinformatics, can you explain what bioinformatics is and what the importance of it is, and then what your course looked into?
So bioinformatics usually refers to the use of computational and analysis tools to capture, integrate, and analyze biological data. And so cancer bioinformatics as a whole refers to understanding the data around cancer that we obtain from online data sets and try to make sense of it to help future studies. And that is exactly what we did.
Great. And then what made you seek out CCIR, and what attracted you to this course in particular?
So my family has two generations of doctors living under the same roof. So biology has always held a strong sort of appeal towards me. And I'm also interested in knowing how the cell works and why some organisms are the way they are. So when I was choosing between the courses, the cancer program really stood out both to me and my dad, because cancer itself is a phenomenon that occurs at the cellular level. And in understanding cancer, I could bring myself closer to the working of the cell as well as the body's DNA, which is the blueprint for life. And cancer is also a phenomenon of mutations in the cell. Well, normally functioning cells go haywire and they start multiplying without any control. And I was keen to see how that happened, how it worked, how it could be corrected.
Great. And then did you have any experience with bioinformatics before you joined CCIR?
It's completely
No, you didn't. Okay. So can you tell people what it was like with this program? You hadn't had experience with it before. How was that for you?
Initially, it was overwhelming because when we were given assignments to read, the biological jargon that was used, I couldn't make head or tails out of it. But over time, as I became more accommodated to both my professor and my TA, I could ask them for help. Over time, I just became comfortable. That's it. Yeah.
Great. And then now we will talk a little bit about your specific research project. So can you tell us what the title of it was and then what you were looking into?
The title of my project was a Bioinformatics Analysis of the Transcription Interactions of Mutant P53. So what that basically means is that P53 is a tumor suppressor gene. That means it prevents cancers. And it is also known as the guardian of the genome because it plays an important role in stopping cell division when it finds some inaccuracies in the cell, so that these mutations cannot be passed on to the daughter cells. However, one interesting feature is that upon getting mutated, this seemingly innocent tumor suppressive P53 now starts acting like an oncogene, that is a gene that promotes cancers. And I was interested to see how that worked out. And since P53 is a transcription factor, that means it enables the transcription of other oncogenes, of other genes, downstream genes, in order to carry out its functions. So I figured that's what mutant P53 does as well. So I just tried to focus on what were the types of genes it could have activated in order to aid tumorigenesis.
Interesting. And then in terms of your research methods and kind of data analysis process, Can you walk us through what type of research you conducted, how you found the data, how you analyzed it?
The data was mainly obtained from online databases as well as literature surveys, and whatnot. And like I analyzed it with the help of my mentor. She helped me go through it, helped me make sense of the data, helped me discard what was not relevant to my research project, and in the end, got fruitful results.
And what had you had experience doing data analysis in this way before? Sorry, have you had experience doing data analysis in this way before? So like in a research project? Okay. Can you explain to us like what was that experience like for you, how your mentor helped you through that?
And my mentor was really helpful because every single time we had a lecture, she would introduce a new sort of online database or an online tool to us. And specifically for me, I chose gene B53. So she would help me understand how I could integrate that particular website and use that data for my particular own gene. And she used examples, she cited different websites and sources. Yeah, she was really helpful.
Great. And then in terms of the findings of your research paper, was there anything that maybe you weren't expecting to find or that surprised you? And can you tell us what the significance of your findings is?
What I found was that in breast cancer with P53 mutations, there is high overexpression of genes that help in cancer progression. And these genes, surprisingly, these genes also have the same transcription associations with P53. So that led me to the assumption that perhaps these genes are activated by P53. If found to exist, these interactions could provide a better stage for new drugs like PROTACs. PROTACs are proteolysis targeting chimeras. They target the protein and completely degrade it instead of rather than just inhibiting its function. And they are really effective in these kinds of situations. So, if found to exist, these drug interactions could set the stage for new drugs, these PROTACs, as well as drugs, PROTACs, as well as scope of further study in how mutant P53 works.
Great. And then have you had any experience with the publication process yet?
No, not yet. I mean, I got the go through by my mentor, but I yet to send it to the CCIR Academy with some staff for the evaluation report.
Okay.
So
and then just sorry?
I'm just waiting to like polish a bit of it and then I'll send it.
Okay, great. And then now we'll shift to discussing just your general experience with the program. So firstly, can you just explain to us your general experience with the program, what it was like for you doing a high-level research project like this as a high school student?
For one, it was much more than a typical teacher-student setup. So Dr. Akman and Dr. Tanner both provided me their individual support and attention. They helped me put forward my own opinions, my own queries. They helped me figure out what I should focus my research on. And also the quality of the content that was being taught, like quite surpassed high school teaching. My dad's a doctor, so when I told him that I was being taught how DNA is denatured, he said that he wasn't even taught that in college. That's kind of tricky.
Yeah, that is really great. And especially in a field like bioinformatics, which seems like it's just taking off with popularity in the last few years. It's great that you get that experience that, like you mentioned, most university students don't even get and you're in high school. And then secondly, how would you compare your learning experience? So both in kind of the learning portion of the course and your experience doing research, how would you compare that with courses that you've taken in high school?
One thing I noticed is that CCIR, I had the freedom to go through what I wanted to do. Whereas in regular high school programs and projects, we are simply given a rigid format to stick to. I didn't find that here. Another thing is that in a regular high school setup, there's 40 to 50 students and one teacher handling them all. But here, because it was so highly selective, I only had 5 students in my course. So, so my mentor, she could focus on all of us individually, and that really helped boost my own learning experience.
That's great. And then I think the first thing that you mentioned is really important too. And especially with just for everyone watching, with both programs, the one-on-one program and the Future Scholars program, we really like to emphasize the aspect of mentorship. So as you mentioned, rather than just the traditional teacher role, the mentors are there to just guide the students in their own thinking journey, in their own research process, help them get through that. But the student is really driving what they want to delve into, which is really great. So can you tell me your experience of how your mentor and your TA helped kind of move you through the first teaching half of the course into your research project and how they helped you learn how to research and how to do the data analysis and all of that?
So during the first 6 weeks, when we were just building up our foundation, they were just giving us the basic information required to set the stage for future studies. And during that time, when I first chose P53 as my gene of interest, I knew that it was the most extensively studied gene, and yet I had my doubts as to whether I would be able to find something new. And my mentor really helped me push past that phase by providing me support. She came up with new databases I could consult, with new omics data I could integrate into it, proteomics, genomics, transcriptomics, and both my mentor and my teacher, both of them helped me kind of they both of them helped me they helped me structure my data, helped me filter through what I found. They also helped me kind of make sense out of it.
Great. And then the last question, or one of the few last questions, the second thing you had mentioned previously is about the small groups of students. So can you tell us what it was like working with students from all around the world? And in this small group, did you enjoy that? Yeah, tell us about working with the other students.
For one, having only used a handful of students in my course meant that I was constantly on guard because I knew that my mentor could see me that. That helped me focus on what was being taught, and also looking at the other students who were trying to show their own interest in the field, trying to ask their own questions, discussing with both the mentor and me and all of us. That meant that they were trying as well. That also showed that we are not alone in this unknown field.
Yeah, that's great. Especially, I think sometimes for students who have these really niche interests, it can be a little bit isolating in the traditional high school setting. So it's really great that you were able to be around students that have your same interests, have your same drive and passion for the subject. So the last few questions I have for you, the first one is, has this course given you any insights into your future? Do you think it's solidified that this is maybe the field you want to go into as a career?
Yeah. So when I first started this, I always knew that I wanted to focus on biology. Now, as we have moved through this course and I got to know more about the working of the cellular systems, and I realized that I probably do want to do this. And like I tell everyone who's asked me this question, I prefer to be on the Darwinian side of things. So I'm currently looking at marine sciences as a possible career option. And I believe that by understanding the DNA, by understanding our genome, and by understanding the cell, this is what we one step closer to fulfilling that.
That's great. And then my last question for you, what would you say to someone who asked you what the value of doing a project or a program like this is, especially as a high school student?
But one, it taught me the ability to critically think at things, critically reason, and the ability to adapt. Because initially my courses ended at 10:30 in the night. That meant I had to sacrifice some part of my sleep schedule. And along with that, I had my school burdens. So I had to find time for both my homework and my research part. Then there's a matter of reasoning and critically thinking at things. looking at things. So when we are given a new set of data, it's completely scattered and sporadic. To look at it questioningly, to see what kind of story it holds. That's something I couldn't get anywhere else.
That's great. So that's it for our student spotlight interview. Thank you so much for chatting with me, and congratulations again on your nomination to our series.